A founding idea of synthetic biology is the objectification of specific DNA sequences as functional primitives that are useful for design. We call these parts, and there are different kinds of parts such as coding sequences, promoters, RNAs, and the like. In some cases, parts correspond to specific molecules that are encoded by the sequence, such as a protein coding sequence and RNAs. In other cases, they correspond to regions of a DNA that proteins act upon with consistent effects, such as promoters and origins of replication. We can simplify the complex behavior of the cell in terms of the central dogma. There is the DNA, which can be transcribed into mRNA, or functional RNA molecules, and the mRNAs can be translated into protein. The sequence of the mRNA is identical to that of the DNA exchanging T for U. The protein sequence is determined through simple rules from the sequence of the mRNA read 3 bases at a time. Then there are the metabolites of the cell that are made through the action of proteins. This includes the primary metabolites that make up processes like the Krebs cycle, but also the membrane and various polymeric materials like peptidoglycan in the periplasm of E. coli. The part concept captures the fact that all genetic information is interpreted according to this model, and the only way to change the persistent behavior of a cell is to make changes to the DNA that underlie these processes. Thus, to change the chemical composition of the cell, we encode DNAs that result in a set of biochemical interactions that ultimately alter the physical state of the cell and its behavior. There are many permutations of interactions that can occur between these encoded components. For example, a protein might interact with a metabolite. A protein might convert one biochemical to another, in which case we'd call it an enzyme. Alternatively, a protein might just bind to the biochemical. These interactions may be quite complex. For example, an enzyme might catalyze a reaction on one molecule while being inhibited by another small molecule. You'd call that allostery. There can also be interactions between a metabolite and an mRNA. For example, riboswitches are RNA elements that bind small molecules and change either the transcription of the gene or translation of its mRNAs. It is fairly rare for small molecules to interact with DNA. However, there are examples of chemicals that will do so. Several natural products, including the anti-tumor agent calicheamycin, binds DNA and undergoes a chemical reaction with it, resulting in strand cleavage and cell death. Beyond interactions with metabolites, proteins may interact with other proteins. Two proteins might bind to one another, forming a complex. That interaction may be inhibitory to one of the partners, or as is often the case, two proteins must come together to give rise to some new molecular function. In many cases, many proteins come together to form a large complex that gives rise to the function. Also, an enzyme might catalyze a reaction that results in a chemical modification of a protein, such as phosphorylation of tyrosine residues by a tyrosine kinase. Proteins may also interact with RNAs. For example, RNase P is an enzyme that does reactions on RNAs composed of a complex of an RNA and a protein. Some proteins bind to specific sequences of an mRNA. The ribosome itself is a large complex of RNAs and proteins that interact with small molecules and RNAs to generate proteins. Proteins also interact with DNAs. They may bind to regions near a promoter and repress transcription. Transcription itself is performed by proteins that bind to DNA and catalyze the formation of RNA molecules. Proteins mediate the processes of replication, packaging of phage and virus particles, formation of transposomes, and so forth, all due to the action of protein-DNA interactions. It is fairly rare for RNA to interact with DNAs, but there are some examples. For example, there are RNA enzymes that can cleave DNA. Thus, when we encode a part, we are describing a functional element in the cell composed of either a DNA, RNA, or a protein. At the highest level, we can distinguish types of parts by whether they implement their specific molecular function at the DNA, RNA, or protein stage of the central dogma.